So in your DNA, there are two different flavors. The first flavor are genes. Now genes, you may have heard of them. They're not like these cool bad boys here. They are sequences of DNA that encode for proteins. And proteins make up the machinery and all the building blocks of your cells. They help life stay alive. Now, the reason why your brain cells look different from your heart cells is because they have different proteins, which means that they're using different genes. So the question is, how are these genes being turned on and off during development? So that's where the other flavor of DNA comes in, and these are called regulatory elements. In this lab, we study a specific regulatory element called enhancers. Now, enhancers, based off of their sequence of DNA, determines where a gene will be turned on and where a gene will be turned off. And that's about all biologists truly understand about enhancers. There's many things that are confusing about them. And we work using flies as our model organism to try to understand how flies develop by looking at their enhancers. So some cool things that can come out of this research are, one, being that we have a stronger understanding of how our genetic code works because our body is filled with enhancers and we don't know how they work. The second thing is different diseases and cancers are often caused by mutations not in the genes but in the regulatory elements like enhancers. Finally, there's also different forms of biotechnology, medicine, and many things that we don't even know about yet that could one day become very important because of this research. Research was really important to me. I joined this research lab about 3 years ago, and during my time here I learned a lot of new things. I would say that in fact I learned more than I ever could have possibly thought I could learn in 1 year in the lab than I did in the classroom. We also learned how to read scientific papers, and that was mind-opening to understand all the different things that are going on in the scientific community and really seeing the scientific method in action. So we'd read about it in papers, then I would go and actually use the scientific method. We'd do a protocol or an experiment, and we don't know what's going to be the outcome or the answer to the question. Versus when you work in a student lab, you already know what the answer is going to be because that's how they can replicate the experiment. So that was interesting, the fact that in research, you cannot go on Google and figure out the answer, because I am the one that has to figure out the answer. And for me, that's just really exciting.